Hey guys, and welcome to today's topic, which is focusing on Pfizer-BioNTech Covid-19 vaccine. So in this series, you will be able to understand why mRNA was chosen, how it works in the body, some clinical trials data explained, why it is safe for ethnic minority groups, advising pregnancy, breastfeeding, and immunosuppressed individuals. Also, its side effects as well as contraindications, plus some of your questions surrounding this vaccine explained. Please, due to the length, I will need to split into different parts. So this is part one. Thanks a lot to all the people that have already subscribed, but if you haven't done so yet, please kindly do so now, and also hit the notification bell so that every single time I upload my health related videos, you will be informed. So without further ado, let's make a start. First of all, let's remind ourselves of how COVID-19 is spread and the symptoms. COVID-19 can be spread through droplets breathed out from the nose or mouth, particularly during speaking or coughing. It can also be contracted by touching the eyes, nose, and mouth after contact with contaminated objects and surfaces. So please, it's important to continue to adhere with government measures to combat the spread of this deadly disease, like wearing face coverings, washing hands regularly and for at least 20 seconds with soap, practicing social distancing, and regularly wiping frequently touched surfaces. After the initial exposure, patients typically develop symptoms within 5 to 6 days, which is the incubation period, although about 20% of patients remain asymptomatic throughout the infection. The main symptoms of COVID-19 are a high temperature, meaning that you feel hot to touch on the chest or back, a near continuous cough. This means coughing a lot for more than an hour or 3 or more coughing episodes in 24 hours, a loss or change to signs of smell or taste. According to the data on WHO website, there are at least 7 vaccines that had been rolled out in different countries at the moment. Also, there are currently 70 vaccines in different clinical phases for 1 to 3 and about 181 potential candidates which have not even reached clinical phase yet. Over 175 million vaccines have been administered globally. Please know that these figures are constantly changing. Now, we need to understand more about this mRNA, as there has been a lot of misinformation lately surrounding this kind of vaccine. mRNA therapy has been in the pipeline since at least 1990, but there is no urgency for its use due to concerns about 1. mRNA instability, because it can easily be degraded by enzymes in the body. 2. It results in high innate immunogenicity, meaning that it can activate the immune system much more than needed, and as a result, this will increase the side effects experienced. 3. Inefficient delivery system, because it is a very large and heavy molecule, it cannot reach the site of action efficiently, as well as because it's very unstable. In 2005, the mRNA was modified so that it can evade the immune system detection. Ever since then, the mRNA had been used to fight against different types of cancers such as melanoma, and the great thing is that this type of therapy can be personalized, but they are all still undergoing clinical trials. Also, there had been ongoing clinical trials in humans for mRNA vaccines against rabies, influenza, Zika, and cytomegalovirus. While tolerability may have been an issue, safety wasn't in all the research studies carried out so far. mRNA vaccines against COVID-19 are a new therapy but not unknown. There had been a lot of work and information about this kind of therapy through various years of research, so we shouldn't be surprised that it has been the first to be approved after its success in clinical trials with good safety and effectiveness profile. Let's be honest, for COVID-19 disease, which came with a very high transmissible rate with increased morbidity and mortality rates like no other, we can't physically wait for 10 to 15 years for a vaccine to be developed, as this will have much more devastating effect on the global population, which will be worse than what we see now. Therefore, a drastic measure to contain this virus is needful more than ever before. This made Pfizer to team up with the German biotechnology company called BioNTech to create a much faster approach using the mRNA. This is because BioNTech have significantly advanced the technology of mRNA through their work in cancer treatment. mRNA medicine carries a set of instructions, hence the word messenger, and these instructions there are cells in the body to make proteins to prevent or fight a disease. We know that the COVID-19 virus has a spike protein on its surface, which it uses to gain entry to cells. So this vaccine works by producing this particular type of protein. Because mRNA vaccine can be constructed very quickly using only the virus genetic code. Hence, the use of this technology is needed due to the global impact of this virus. It is a speedy approach in the face of the urgency of the pandemic compared to the use of conventional methods. Before I carry on, let's look at the differences between mRNA and conventional methods, as this will help to understand why this vaccine was very speedy. In the conventional method, a medium, for instance eggs, are needed to multiply the virus, which can take several years to develop, whereas the DNA of the spike protein can easily be developed in the lab, which will now be transcribed to the mRNA, and this process can take just a week. In conventional methods, large amounts of the viruses are needed. With mRNA vaccine, no virus is needed to make the vaccine. Only small quantities of viruses are needed for gene sequencing purposes and vaccine testing. In the conventional method, the right strain of the problematic virus is needed, and a lot of them for it to be successful. And due to the massive production, a local outbreak of the disease could occur, whereas the mRNA of the problematic spike protein is synthesized in the lab. The conventional methods are expensive and very difficult processes are undergone, but the mRNA is cheaper and more straightforward, mainly because it is being synthesized. Also, in the mRNA, there is no need to extract and purify cultivated viruses with all the precautions required to avoid leaving potential toxic product in the vaccine. There is also no need to inactivate or treat them to isolate the S protein compared to conventional methods. In terms of how they work, in the conventional method, once it's injected, the inactivated or killed virus will act on the cells, which will start to create antibodies and also activate other immune system cells. Whereas the mRNA, once injected, it tells the body to produce the S protein required, and this protein will be displayed on the cells, which will now trigger the immune system to start creating antibodies and also activate other immune system cells.